Good morning, everyone. Today I will be discussing Bacillus anthracis and how it interacts with its environment and its hosts. Let's get started. Bacillus anthracis is a rod-shaped, gram-positive bacterium which is well known for causing the potentially life-threatening disease anthrax. Highly contagious, its severity ranges from mild to fatal depending on how it was transmitted, through contact or ingestion. It is also capable of producing numerous virulence factors which could further increase the severity of the infection. Bacillus anthracis is known to exist in multiple habitats and forms, commonly found in soil where it forms spores that can remain viable for long periods of time and are especially resistant to extreme environmental conditions. Additionally, it is also found in the intestines of some animals, particularly herbivores such as cattle and sheep, which serve both as a source of infection as well as a potential reservoir. Bacillus anthracis is a rod-shaped bacterium, measuring 1 to 1.5 microns wide and 3 to 5 microns in length. Characteristically, it has a unique capsule made up of a protein called poly-D-glutamic acid, which helps protect it from destruction by its host's immune system. Bacillus anthracis is a strain of bacteria that can display distinct cultural characteristics in a laboratory setting. Blood agar can produce large, grayish-white colonies with a medusa head look, and similar colonies can be found on McConkey agar and nutrient agar. Therefore, it is essential to note these particular cultural qualities to accurately identify this bacterial strain. B. Anthracis is a facultative anaerobe with catalase-positive properties and oxidase-negative qualities. Additionally, it produces a range of enzymes including proteases, lipases, and hemolysins, enabling it to break down proteins, lipids, and red blood cells. Bacillus anthracis is capable of infecting humans, cattle, sheep, horses, and pigs. It can present itself in 3 forms: cutaneous, gastrointestinal, or inhalational anthrax. Understanding its potential impact on public health requires knowledge of its host range. Bacillus anthracis, the bacterium that causes anthrax, is very dangerous due to its ability to produce a variety of molecules and proteins that can harm tissue and suppress the immune system. These virulence factors include the capsule that protects it from phagocytosis, as well as the anthrax toxin, consisting of 3 proteins that interfere with cellular signaling, leading to tissue destruction. Other proteins and enzymes produced by the bacteria can also help it avoid the host's immune response. Bacillus anthracis is often susceptible to antibiotics such as penicillin, tetracycline, and fluoroquinolones. However, strains have developed resistance to others like erythromycin and clindamycin. Several mechanisms can cause this, including gene mutation or production of an enzyme that can inactivate the antibiotic. Additionally, some strains of Bacillus anthracis can transfer resistance genes to other bacterial species, meaning resistance can spread quickly. Anthrax is a rare disease in humans, but if not treated correctly, it can have serious consequences. Contracting the disease usually happens through contact with infected animals or animal products such as wool or hides. Inhalational anthrax is less common, but it can occur due to exposure to contaminated soil and animal products. Being aware of the risks and taking preventive measures is essential to avoid infection. Anthrax is a dangerous bacterial infection that can manifest in 3 different forms. Cutaneous anthrax, which is marked by the appearance of sores on exposed skin. Gastrointestinal anthrax, which can cause nausea, vomiting, and abdominal pain that can lead to severe complications. And inhalational anthrax, which can lead to life-threatening respiratory failure. All forms of anthrax are caused by the production of the anthrax toxin by the bacterium, which causes tissue damage and disrupts cellular signaling, resulting in potentially fatal consequences if not treated in time. The host's immune system is a complex and multifaceted defense mechanism against Bacillus anthracis. The bacteria possess a capsule, as well as a range of enzymes and other proteins that allow it to evade our body's natural responses. In addition to providing protection against the capsule and other surface molecules, our immune system also uses both innate and adaptive immune mechanisms to prevent infection. Diagnosis of anthrax involves the observation of clinical symptoms and laboratory testing. Serological tests like blood cultures and culturing bacterium from clinical specimens like blood or tissue samples are used. This approach helps to ensure an accurate diagnosis. Hygiene and proper handling of animal products are essential to avoid anthrax. A highly effective vaccine is suggested for those in high-risk areas, for example veterinary clinics, animal handlers, and laboratories. To cut the chances of exposure, one should take precautions, like wearing protective clothing and following safety guidelines when dealing with animal products. Anthrax is a serious health threat that requires prompt and proper treatment. A combination of antibiotics and supportive care is recommended. Penicillin, tetracycline, and ciprofloxacin are highly effective for treating Bacillus anthracis, but treatment should be started right away to avoid the illness becoming more severe. Oxygen therapy and fluid resuscitation may be needed in more serious cases of anthrax. Thank you for your attention.